A very good morning to all of you. Myself, Dr. Varun Narayan Mishra, and today we are going to discuss about the fermentation process, in which first we will see the historical development of this process, then components of different fermenters and the types of fermentation. So it's an important topic in our ecosystem, in our ecological process of the earth. This fermentation process is very important. First, I would like to start with the definition of the fermentation. So it's a metabolic process that produces the chemical changes in the organic substrate. Okay, basically in our Earth, on the Earth surface, or you can say of the entire globe, different organic materials are there. So the process of the chemical changes in the organic substrate through the action of enzymes is known as fermentation. Okay, when we split the organic substrates into different components, or into different other substrates, into different other materials using some enzymes. Enzyme is a kind of chemical that is used to change the chemical properties of any organic material. So fermentation is a metabolic process that produces the chemical changes in the organic substrates through the action of enzymes. In biochemistry, it is defined as the extraction of energy from the carbohydrates in the absence of oxygen. As we know that the carbohydrate is the major source of energy, but it is not directly available. So we need to extract the energy from the carbohydrates. So for this, we need to perform some metabolic process. We need to perform a fermentation process that extracts the energy from the carbohydrates. Okay, this energy is very important for human life as well as different plants, different other animals, because each and every living organisms on the earth, they require some source of energy, and carbohydrate is one of the major source of energy for all the living things on the earth surface. Okay, but if there is no oxygen, so how to extract that carbohydrate, how we can convert that carbohydrate into energy? For that we use some enzymes. Enzyme is a kind of chemical material that will be helpful for extracting the energy from the carbohydrates using some chemical reactions. Okay, and fermentation in food processing is the process of converting the carbohydrates to the alcohol or organic acids using microorganisms, yeast or bacteria under anaerobic condition. Okay, and another definition of fermentation will be defined as the conversion of carbohydrates to the alcohol. Means when we are going to extract the energy from carbohydrates, so during this chemical process, alcohol is a byproduct. Okay, when we extract the energy from the carbohydrate, so a new material or new component will be available as a form of byproduct, which is known as alcohol. Okay, and it can be done using some bacteria. Okay, for this process we can use some bacteria. These bacteria are very much useful for initiating the fermentation process, and these bacteria are not harmful to the human bodies. Fermentation usually implies the desirable action of the microorganisms. In microorganisms, like microorganisms means it is talking about the cells in the body of the human. Fermentation is the primarily means of producing the abionize triphosphate ATP by the degradation of the organic nutrients, and the science of the fermentation is known as zymology. Okay, so fermentation is a process of the conversion of carbohydrates into energy using some kind of enzymes. That enzyme may be any kind of bacteria, it may be any kind of chemicals. Okay, and the entire process is known as fermentation, and the science of the study of fermentation process is known as zymology. Okay, so the historical development of this fermentation process is started in early 30,000 years old. Since the ancient time, humans have exploited the fermentation process. The earliest archaeological evidence of the fermentation is around 13,000 years old residues of a beer found in a cave near the Haifa in the Israel. Okay, so this process is very ancient. Uh, for the long period of time, the humans are started using this fermentation process to convert a material into another material using some processes. Okay, another early alcoholic drink made from the fruit, rice, and honey dates from 7,000 to 66,000 BC in the Neolithic Chinese village of the Jiao, and the wine making dates for the 6,000 BC in the Georgia. So you see a lot of historical things, historical evidences are available for this fermentation process. Okay, so there is a strong evidence that the people were fermenting the alcoholic drinks in the Babylon 3000 BC, ancient Egypt in 3150 BC, and pre-Hispanic Mexico 2000 BC, and the Sudan in around 1500 BC. So these are some historical evidences which is present, and it shows that the humans have started using this fermentation process for converting a material into another material, for extracting energy from different organic materials for their use. And most oldest evidence which is found is around 13,000 year old. Okay, so traditional fermentation technology, the recent developments in the fermentation process is more than 3,000 year old in India, as mentioned in the ancient literature. A sweet substance known as soma juice that is prepared by the Vedic Aryans is supposed to be the first product of the fermentation in the India. Okay, the Rig Veda, that is around 1500 BC, shows that the fermentation technology took its first step in the connection with the preparation of som juice in India. Som juice means you can call it as alcohol. Okay, okay. So there is also another drink known as sura, that is considered a wine or beer. Okay? In the ancient time, the wine and beer is known as sura. So in India, the process of the sura, the preparation of the sura has been started during the using the fermentation process. So it is believed that the acetic fermentation was also known to India since the early time. Curd is another very popular fermentation product which is described in the Rig Veda. Okay, it is still remembered a popular food and the technology for curdling milk that is found in another number of texts associated with Yajurveda. Okay, so curd is a byproduct of fermentation. That is the process in which the milk will be converted. The milk will be converted into the curd using some chemical process, using some bacteria. Okay, so in the beginning, the fermentation was mainly associated with the preparation of spiritual drinks, but later on it was used for another purposes also. Okay, so these are some historical facts, and you see the fermentation process is very old. This technology, this process is very old, and the humans started using this fermentation process for a long time ago. Okay, so this word fermentation is derived in the recent time. Okay, initially it was not known as a fermentation, but in the recent history, in the recent time, the term fermentation is derived from the Latin word which is known as fermentum, that stands for the boiling. The French chemist Louis Pasteur founded the zymology. It's a study of fermentation. When 1856 he connected yeast to the fermentation process. So that the term gymology was started in 1856 by the French chemist. He was very famous, Louis Pasteur. He developed a branch of the science, zymology, which study the fermentation process. The food fermentation is the conversion of sugars and the other carbohydrates into alcohol, organic acids, and the carbon dioxide. All three products have showed their importance in the human use. Okay, so the basically in the fermentation process, we convert one chemical or we convert one product into another product, and as we know that oxygen is a basic element for all the products in on the earth surface. Okay, so the fermentation is used to convert that material into another material, that is a conversion of sugars and the other carbohydrates into alcohol, some organic acids, and the carbon dioxide. The production of alcohol is made use of when fruit juice are converted into the wine, or when grains are made into the beer, and when food reaches the starch, such as potatoes. Potatoes are fermented and then distilled to make spirits such as gin and vodka. Okay, so the entire process of fermentation is now very useful for the preparation of different kinds of vodka and wine, beers, and other byproducts. Okay, fermentation is one of the oldest technologies used for the food preservation. It is a process of digesting certain substances that leads to the chemical conversion of organic substance into simpler compounds. Okay, over the centuries, it has evolved, that has been refined and diversified. Today, a variety of fermented product is available in the market. A wide range of raw material is used as the substrates in the fermentation, and resulted and products derived from fermentation are major constituents of the human diet all over the world. Okay, the fermentation system may be the liquid, which is also known as submerged, or solid state, also known as surface. Most fermenters, the product which is used for the fermentation process is known as fermenters. Most of the fermenters used in the industry are of submerged type, because it is the fermenter, it is a kind of fermenter, they saves the space and is more susceptible to the engineering control and the design. So there is much work still needed to done to identify the best fermentation technique for each bioactive compound. So basically, when we talk about the fermentation, so it is basically applied for the bioactive compounds. It is basically applied for a compound which is having some amount of oxygen, and or you can say the compounds of the oxygen. Okay, so this is the systematic representation of the common types of fermentation, different kinds of microorganisms which is involved in this fermentation process, and what will be the end product, or what will be the byproduct of the different kinds of fermentation process. So basically, fermentation process is of four types. First one is alcoholic fermentation, lactic acid fermentation, acetic acid fermentation, alkaline fermentation. Okay, and this these are the microorganisms which is involved in the process. For alcoholic fermentation, we use yeast. For lactic fermentation, basically it's a kind of acid bacteria. Then for acidic fermentation, they also use some acidic acid bacteria. And alkaline fermentation is mainly for Bacillus species. These are some kind of bacteria. So the byproduct or the end product of the alcoholic fermentation is alcoholic beverages. Then the byproduct of lactic acid fermentation is lactic acid. Byproduct of acetic acid fermentation is a kind of acetic acid, and alkaline fermentation will be converted in the form of amino acids. So this is the representation of different types of fermentation, different kinds of processes for different fermentation, and what kind of microorganisms is involved in the fermentation process, and what will be the end product of that fermentation process. Okay, so this is the production of the fermented food by the bacteria and yeast. Okay, so glucose will be converted into pyruvate, then it will be converted into acid, lactic acid, that is a soya sauce, then lactic acid in the form of cheese, beer, ethanol, and the carbon dioxide as a will be used as a uh, substrate for the component for the bread. So these are the production of the different fermented foods by the use of bacteria and yeast, and all are needed to have some kind of energy source, that may be glucose or carbohydrate, and then it will be converted into different products using different fermentation process. Okay, so this is a design of an ideal fermenter. Fermenter is a instrument. Fermenter are a kind of you can say the apparatus, is a setup that will be used for the conversion of one product into another product through the fermentation process. So you see it is having multiple components, it is having different parts, motor, then pH control reservoir is used to control the pH level value. Then there is uh, a part which will be used to supply the steam. Okay, then penicillium intel is there. And then there is a option there for out for releasing the steam. Then cold water jacket is there. The temperature recorded and cold water pump control is there to control the temperature. And cold water inlet is used to supply the cold water into the instrument, into the setup. And there is a air filter. The air filter is used to supply the air, because air will be used as a uh, substrate for initiating this entire process using some air filter. Air filter uses this to convert the air, and it will be used for this fermentation process. Okay. The various components of an ideal fermenter are the top plate that will be cover made of steel. Then there is another part which is known as clamp. Clamp is a top plate compressed onto vessel using clamp. These are some components. And fermenter is an instrument where the fermentation process occurred. For initiating any fermentation process, we need a setup, we need an instrument, or you can say we need a proper apparatus that is known as fermenter. Okay. Third part of a fermenter is a seal. It separates the top plate from the vessel to prevent the air leakage. Then vessel, it's a kind of glass that is jacketed the steel with the ports of the various outputs. Drive motor, drive shaft, marine impeller, baffles, okay, sparger, exit gas cooler, inculation needle, feed pump, okay. These are some parts of the ideal fermenter, okay. Then the monitoring and the controlling part of the fermenters, how to control different uh, pr- uh, parts, how to control the proportion of different materials in the fermentation. We need to have PT100. these are the these are used to control the temperature. It is a temperature sensor. Foam probe is kept above the medium level to sense the foam formation. pH electrode, it senses the pH level and control the pH level value. O2 sensor O2 sensor monitors the dissolution of the oxygen. It controls, it senses or monitors the oxygen level in the fermenter. Heater pad directly hits the medium. Cold finger, it after the direct heating, it is used to cool the vessel contents, that is closed coil pipe to pass cool water. Rot- Rotameter, that is a variable air meter. Pressure valve is attached to the rotate meter for safer operation. Air pump is used for the supply of air. And peristaltic pump is used to pump the medium of different acids, bases, and the antifoam. These are some monitoring and control parts of any fermenter. Okay, so these are the beneficial properties of fermentation process. These are the properties of any fermenter. Like it should have oxygen control, temperature control. There should be the optimum value of the pH. Then correct species and strains is required to be used during the fermentation process. And we need to perform the qualitative and quantitative active substances. Whatever the substances we are using in the fermentation process, we need to study the qualitative and quantitative properties of different substances. Cold pure culture, it should be fully controlled continuous fermentation, low risk of the contamination, and the optimal nutrients should be required for any kind of ideal fermentation process. So these are some beneficial properties of the fermentation process, or you can say these are the uh, characteristics of an ideal fermenter which can be used for initiating any fermentation process. Okay. Now moving to the next slide. The fermentation is a chemical transformation of different organic materials into different complexes, different kinds of compounds. That may be because of enzymes, that may be because of different microorganisms such as bacteria, yeast, and other components. In other words, fermentation is the technique of the biological conversion of complex substrates into simple compound by various microorganisms. So it may be classified into two types: solid state fermentation SSF, submerged fermentation SMF. So the development of this fermentation techniques has led to the industrial level production of different kinds of bioactive compounds such as antibiotics, pigments, antioxidants, anti-tumor agent, biosurfactants, peptides, and the metabolism exhibited by the microorganism is different in the SSF and SMF. So I know now we will see what is the SSF and what is SMF and how they work and which one is better than each other. Okay, so this is the comparative analysis of two methods of the fermentation: submerged fermentation and solid state fermentation. Okay, so submerged fermentation may be carried out as a batch of continuous process, whereas the solid state fermentation may be carried out as a batch, as a part. Its submerged fermentation is having a medium which is added in the large vessel, whereas in solid state fermentation, medium is added in flat vessel or the trays. Surface area to volume height ratio is very less, whereas the surface area to volume height ratio is very high in case of SSF, that is solid state fermentation. Okay, and fifth point is column is usually in liquid form, whereas in there is a uh, in column it is usually spread on the surface of the media. So these are some comparative analysis of two different kinds of fermentation process, and you see that both are different from each other in different aspects. And here you see in the submerged fermentation, power consumption is very high, whereas in solid state fermentation, the consumption of power is very low. It is less power consuming technique. The controlling parameters like temperature, pH is very easy in submerged fermentation, whereas the controlling factors like temperature, pH is very difficult to control in the solid state fermentation. Okay, in submerged fermentation, it is required to have less manpower, but in solid state fermentation, we need to have more manpower. It required more manpower. Okay, so substrate used, more what kind of materials or what kind of substrates are used in both the processes, that is submerged fermentation or solid state fermentation. So it's a comparative study between the SMF and SSF, and the types of substrates which can be used for those processes. So for SMF, the soluble sugar, okay, molasses, liquid media, fruit and vegetable juices, sewage, wastewater can be used as a substrate, whereas in case of SSF, wheat bran, rice, straw, fruit and vegetable waste, paper pulp, okay, coconut coir and synthetic mediums are can be used as a substrate for the SSF. So these are the comparative study of different kinds of substrates used for both SMF and SSF process. The solid state fermentation SSF, it has been defined as the process that occurred in the absence of free water. It doesn't require any water, and this is the method that used for the production of enzymes that involves the cultivation of microorganisms on a solid substrate such as grains, rice, and the wheat bran. And SSF employs natural raw materials as a carbon source such as cassava, barley, wheat bran, sugar cane, various oil cake like palm cake, soybean cake, groundnut oil cake, fruit pulps, and sawdust and seeds. SSF has been defined as the fermentation process occurring in the absence of free water, and it generally utilizes the solid substrate. It's a biomolecule manufacturing process is used in the food industry, pharmaceutical industry, and these are mostly the metabolitics that is generated by the microorganisms grown on a solid support selected for this purpose. And SSF technology is an alternative to the liquid or submerged fermentation that is used predominantly for the industrial purposes. So why solid state fermentation is better than SMF? Why SSF is better than SMF? So it's very simple and cost effective. It is having less effluent release. It reduces the pollution level. A- Aeration is very easy. It resembles the natural habitats of some fungi and bacteria. SSF utilizes the solid substrates, thus the nutrient-rich waste material can be easily recycled. It These are used as a very slow and stably, so same substrates can be used for longer fermentation process. SSF is best for the fermentation process that involves fungi and other microorganisms that require less moisture content. Okay, so this is the simplest and very cost effective process of the fermentation, and it the process is very slow in the solid fermentation. That's why it can be used for the longer fermentation process, and it generally based on the fungi and different microorganisms. So it is very important, and it can be used for producing the nutrient rich waste material that will be used for different other purposes. Okay, so these are the advantages of solid state fermentation. And this is the solid state fermentation process, how it works, and what are the components of a good fermenter that can be used for the solid state fermentation. It should have some air outlet lead, then there may be some in column module bases, cooling devices should be there, cultivation substrate, air inlet, water supply, water discharge, and the output will be in the form of nutrient rich materials that will be having some properties that will be used for different practices, different applications. So these are the factors which are involved in the SSF process. The entire process having four factors. First, we need to focus on the selection of proper microorganisms, what kind of substrate we are going to use, what is the process of the optimization. It is very important. This is the important factor during the process of SSF. And what kind of isolation and purification products can be found after that process. So selection of microorganism is very important. It is one of the key factor for improving the process, for improved yield of the byproduct. Bacteria, yeast, and other kind of fungi can be used. And filamentous fungi has shown better result growing in the solid substrates of fermentation. And substrate is very important. It's a very important role in the process. So we need to choose a proper suitable substrate during the process of the fermentation. Okay, one is specific substrate which requires suitable value addition and or disposal. The second is for producing a specific product from a suitable substrate. Process optimization is the optimization of physiochemical and biochemical parameters, which include the size, the moisture content, the pH level, the pre-treatment of the substrate, relative humidity, temperature of the incubation. The nutrient supplements such as nitrogen, phosphorus, and different trace elements, trace materials, and supplementation of additional carbon source and inducers. Okay, application of SSF is in various fields ranging from the pharmacology to bioremediation, covering the various aspects of the biodiversity conservation. So these are the applications of solid state fermentation. The production of the industrial enzymes are almost all the known microbial enzymes that can be produced under SSF system. It can be very important industrial applications. It can have important industrial factors like protease, cellulose. Okay, and we can have the production of biopesticides using this SSF. The infamous Bacillus thuringiensis protein can be produced in the large scale in order to address the issue of pest attacks, yield damage. These are the applications of solid state fermentation process. It can be used in food industry for the traditional food fermentation, mushroom production, then bioconversion and byproducts, food activities, food additives. It can be used in agriculture for biocontrol, insecticides, plant growth, and enhancer. It can be used in industries like enzyme production, antibiotic production, organic acid production, fungal metabolites, and ethanol production. These are the applications of solid state fermentation, but still there are some problems in SSF fermentation. Okay, one of the main difficulty in SSF process is to control the temperature during the process. Heat is generated during the metabolic activities of the microorganisms. Since the substrate used has low thermal conductivity, heat removal will be slow. Okay, when the heat generated, that will be cross the beyond, that will be cross a certain level, which will result in product degeneration, and it will affect the growth of the microbe, ultimately ending up in the reduction in the yield and quality of the product. The only problem in the SSF, only problem in the process of solid state fermentation is to control the heat, because the heat releasing capacity in the solid state fermentation is very slow. So it may affect the byproduct, it may affect the quality of your byproduct because of the high temperature in the fermentation process. Okay, submerged fermentation is another type of fermentation process. Okay, it is a technique that will be used for the cultivation of microorganisms in the liquid form, which breaks down the nutrients to release the desired bioactive compounds. In this method, the selected microorganisms are grown in closed vessels that will be used for containing a broth rich in the nutrients and the high concentration of oxygen. So in submerged fermentation, they utilized quite rapidly, hence needs to be constantly replaced or supplemented with the nutrients. Bacteria requires high moisture content or high water activity and are the best suited for the submerged fermentation. Okay, submerged fermentation basically requires some liquid. Okay, so it requires high moisture content and high water activity that will be very much useful for the submerged fermentation. So the applications of SMF, that is submerged fermentation, it utilizes the free flowing liquid substrates such as molasses and the broths. The bioactive compounds are secreted into the fermentation broth. The substrates are utilized quite rapidly. That's why the substrate need to be constantly replaced, supplemented with the nutrients. This fermentation process, the entire technique is best suited for the microorganisms such as bacteria that require high moisture content. And one of the additional advantage of this SMF technique is that the purification of product is very easy. It is primarily used in the extraction of secondary metabolitics that needs to be used in the liquid form. Okay, so this is the process of submerged fermentation. The entire submerged fermentation process is shown here. It should have some media preparation component, then media sterilization, bioprocess control can be done by some programs, some computers. There may be some in-column preparation, cell recovery, product recovery. There may be a component for the waste treatment and the disposal of different solid waste which will be produced during this process. So more than 75% of the industrial enzymes are produced using SMF. One of the major reason is that this technique supports the utilization of genetically modified organisms to a greater extent than the SSF. Okay, and in submerged fermentation process, the accumulation of a variety of intermediate metabolitics results in the lowered enzyme activity and production efficiency. It is based on the research. Certain bioactive compounds that have been found to be produced in the higher quantities in the SSF, whereas other compounds have been extracted using the SMF. So both the processes, when we talk about SMF or SSF, both are having advantages and disadvantages. Both are having some different applications. Both are having different processes. So we need to choose any of the fermentation process as per our requirement. We can use any of the fermentation process depending on the need, depending on the product, what kind of product we need. So we can choose any of the fermentation process, either SSF or SMF. So thank you very much for today's class. And today we have discussed about the fermentation process, then types of the fermentation process, what are the historical developments of the fermentation process, and what are the applications of this fermentation process in different fields like pharmacy. Then it may be used for some industrial activities, industrial applications. It may be used for producing different byproducts, food products. It can be used for in food processing industries, for conserving, for preserving the quality of different materials, different byproducts, food products. Thank you very much for today's class, and we will discuss another topic in next class, that will be the biodegradation. So we will meet again tomorrow in the next class with the next topic. Thank you very much for your patience listening.